Master Sani, Applied Biomedical Research, and I'll be giving the webinar today on the clinical data analysis for GDC. So I'm going to give you a short overview of the clinical data analysis, go through the workflow, which I have slides for it, but I'm primarily going to rely on a demo. So I'm going to give you a live demo of it, and then I'm going to come back and give you some references and user guides and stuff. So just a quick overview of the clinical data analysis. So tool in GDC, it's allowed to specifically allow users to analyze the clinical data at a cohort level in GDC. Users can pick, make a cohort, and then pick clinical fields that they want to analyze visually using various plots like histogram, survival plots, box and whiskers, Q2 plots. We'll go over those in a second. They can also create custom bins and then re-visualize the data in those within those bins. And then there are specific cases where you would want to, once you have analyzed or your data, you may want to create a new cohort out of them. So I'm going to show that. That's a very powerful functionality. So basically, within the analysis, you can create a new cohort and then go through the process, circular process. Also, you should be able to download the visualization plots as SVGs, PNG, and JSON formats, and also the table as TSV format. And you can print the whole thing as PDF. So the clinical data analysis workflow kind of in GDC goes through exploration, and then we create a custom cohort, and then we allow users to pick the cohort within the analysis, and then perform analysis. And we'll go over these 4 steps. And once you perform the analysis, what I just talked about was you can create a new cohort out of it, and then go back into exploration or repository anywhere else within GDC. So we were going to start with the exploration, and then we'll work our way down. Within exploration, we've added a new tab in the last year called Clinical tab, and I'm going to go into detail about that. That is than the previous iterations. That goes into a lot more detail and lets you build a clinical cohort. So with that, we'll go into GDC demo. So here I'm on GDC portal, data portal. I can pick exploration from the menu or from here. Once I go into exploration, I'm seeing my cases, clinical tab, genes, mutations. So I'm seeing them all. I'm going to start with clearing the query and starting from top. The example I want to do is I'm interested in, let's say I'm interested in melanoma. Specifically, I'm interested in the cutaneous melanoma, which is a skin cancer, where it starts in the pigments and cells related to the pigments of the skin. So I'm interested in that. So I'm going to show you how I can do that. So I have there were different ways of going through it. I can start with the primary site, programs, projects, disease types, experimental strategy, and sample types, and various data. In the clinical ones, there's a search box for me. I can start with demographics, gender, race, vital status, cause of death, etc. I can collapse this. I can go into diagnosis, treatments, and then exposure. So there are various ways I can build my cohort using all these different clinical fields. So I was jumping the gun a little bit. I wanted to start with a TCGA study on melanoma. So I'm picking TCGA. And this is just an example because I thought it has some rich data in it for us to explore. And I'm picking the TCGA SKCM study. So as I see, there are 470 cases associated with this, and I'm interested in exploring the clinical data associated with this study. So in order to build my cohort, I'm going to go to Save Edit Case Set and click on that, and gives me an option of Save a New Case Set. Tell me the number of cases. I'm going to click on that, and by default, it created a name which was based on the query. So it gave me a TCGA SKCM as the default name, and I'm fine with that name. So I'm going to save that name. And it's telling me that there was a case set created for it, and I can view it in the manage sets, but I'm interested in analysis. So I'm going to go into analysis. So once I go into analysis, there are various different analysis that we support. The feature I'm going to talk about today primarily is the clinical data analysis, which has a demo feature for you, and which is quick and easy to do, or you can do a select, and I'm going to click on select here. So right now I have one case set that I just created. I'm going to just select that. If there are more than one, then you have a radio button and you can pick one of them that you want to see. So in my case, I'm just picking the one I just created. I just created my cache, everything. So just you have a clean slate to start with. I'm going to run the analysis. And this is the clinical analysis tool dashboard that comes up. It is broken down into all the different analysis that I can run on this side, and demographic, same thing, demographic, which has race, gender, ethnicity, vital status, etc. I can collapse that, diagnosis, which has various diagnosis, staging, grading information, and there are like 59 fields associated with it, various treatment things we capture, and then exposure. So very similar to how the cohort is built. There is also a search button that lets you search because there are lots of fields, and to help you do a quick search. We'll come back to that in a second too when we do a search. On the other side is what we have are cards, clinical cards, and it creates 6 cards for you by default. Overall survival for your cohort, so any cohort you create, and if it has survival data. For example, there are 470 cases we started with in our cohort. There's survival data only for 456. So that's why it's plotting 456, not 470. So if there is data for it, it'll plot it. And then it gives you a quick other cards. The 5 other cards are ethnicity, and you can see they're already set over here. Gender, race, ethnicity, age of diagnosis, and then the last one is primary site. And the colors kind of correspond to the different fields. So if I go back up, you'll see these are blue button switches, and these are blue plots. So it's just to help you remember where you got that card from. Other thing you can do is you can collapse your control, and then you get 3 cards at a time, and you can scroll down. There's 6 cards for you to look at. And this is really helpful. I'll come back to it for printing purposes where you don't want to show, but you know, your criteria or how you got the controls. You can clean that up. So here's my case, the cohort I created, the name of the cohort, 470, and with the 6 default ones. I can go down and switch any of them and create new cards for myself. So, and each of the cards, back to it, each of the cards have these little icons next to it, and it tells you the various features for it. For example, over here, I can download the plot. I can reset the plot. This is a histogram we're viewing. I can switch the ethnicity to a survival plot, where it's plotting the 2. It'll plot the 2, by default, start with the 2 highest ones. So in this case, it's non-Hispanic and not reported, but I'll come back to that in a second. And then I can remove a card. If I don't want to see ethnicity, I can just basically remove it. The next one moves up, and actually I can download the image. When I click on that, it shows me the different various formats that are supported. So the overall survival there is no closer for that, and that stays in as default. And when we're plotting the overall survival, I can hover over it, and it'll start giving me some values. So for example, for this melanoma study, I'm looking at around 5 years, and so around right here, around 5 years, it's showing me that about 59% survival rate at 5 years, and then going down. Now these dashes are too close together, and I can always hold my mouse down and expand on it. And as you can see, the scale changed, and now I can see a lot further. I can, in any plot I can do that. Survival plot, I can do this. So it's a good feature to see. And I can always reset it. So when I click on it, it resets it back. So now what I did was I just showed you the survival. So I closed the ethnicity, so the gender moved up. So let's look at survival plot for gender. So when I click on gender, it's actually mapping the survival for the male and female gender for me. So they're about 60% male in this cohort and about 38% female, and it's doing survival for both of them. And I can do the same thing. I can actually look at it, like for example, at about 14, 15 years survival for expand it a little bit. So for about 15 years is about 33% for female and about 20% for male. Actually, I can also download the data as a TSV format, and it'll show me, the plot I have. This lets me see the cases that make up this 60%, and then for the two. If I click on it open into a new window, it starts plotting those, and it's giving me the actual cases. So I'm going to go back to my analysis. And as you can see, these are the various plots. There's another type of plot that comes as default, and I want to talk about that, which is and same thing, the race works the same way, is age of diagnosis, and those are the numerical data. So these are all categorical data, what I just showed you, where we have a limited set that that submitters, when they submit data to GDC, is using. So it's a controlled vocabulary, whereas the numerical ones is an integer or some kind of a numeric value. Doesn't need to be integer. And it's showing me, in this case, in days. So it's showing me the age of diagnosis in days as default. And it has two other plots, site survival, and it has a box and QQ plot. So the survival is very similar. It's taking, QME survival, and it's plotting the two highest categories. And then I come back to box and whisker, and it's giving me a box and whisker plot and a QQ plot. The box plot is plotting the mean for me, and the median is plotting the first quartile, first quintile, and the third quartile, and then the max values. And then same thing, I can see the values numerically at the bottom. So it's giving me the age is around, minimum age is 15 in this cohort, the maximum age was 90, and then the mean and median are pretty close, really close to each other. And there is a standard deviation of 15. The QQ plot actually takes the median and then splits into two quartiles, the people under 50% and the people above 50%, the cases below 50%, cases above 50%, and plots both of them across the 45-degree angle line. And it tells me, how well the data is distributed. In this case, as you can see, it's fairly well distributed, that, it's almost hugging the 45-degree line. At the top range, it is a little bit wearing off, and same thing at the bottom. It's there are only a few cases that are, around 15 age. So it's a very useful plot to show you the distribution of data. Okay, so I'm going to go back and do a search. So in this case, there, as I said, there are a lot of fields that I can go through them. And basically, I can add cards back, like ethnicity, by switching it or closing it, or switching it off. And I can create and assemble my cards however I want to. So as I said, there are a lot of cards, and I want to go back and do a search. So I want to do a search on stage. So it showed me that in diagnosis, there are a bunch of fields that are stage. So it's showing me 1, 2, 3, 4, 5 as default. There are 7 more. I click on it. It's showing me all 7. So it works on anything you can type on. It will try to find it and do a search on it. So in this case, it's showing me there's it's clinical staging. I might have information. So when I click on it, if that study didn't collect clinical staging, ACGCC, then it will say no data for this field, and it'll show up like this. And you can either close it, or you can switch it off. I'm going to go to pathological stage and check that out. And I see there is pathological stage information. So it's kind of discovery analysis. You're looking through it, and it's telling me the lots of stages information I do have within this cohort. And it's broken down quite a bit. So another feature that the clinical data analysis supports is binning. And this is all categorical data, so I'm going to try to bin it. So when I click on custom bins, it shows me edit bins. So I'm going to click on edit bins, and it's showing me all the values. This is a very, we try to make it into a very user-friendly feature. So I'm going to try to bin my stages together. So I'm going to take all the ones. So I'm just clicking on one, and it'll hold on it. I'm not, there's no other, I just clicked on it, then I'm clicking stage. So I'm looking through for the ones. What else do I have? Okay. So I'm going to try to group these together. Click on the group button, and I can give it a name at this point. I can do stage, and if I typed lowercase I, it's told me there's already a group name I, so there's already a category I, so I can't use that. So I'm going to just use uppercase and hit enter. So I basically grouped 3. So I'm going to try to do the same thing with the stage 2s. Stage 3, and I just noticed that I missed a few of them. For example, this was 2. So no worries, you can just click on this, and do a group, and it'll move it to that stage. Same thing for, I missed 3A. So I'm going to click on this. And there, I got them. So I'm going to leave stage 0 as is and stage 4 as is, and not reporting. So I'm going to do a save bins and see what it changes. So as you can see, now it's trying to plot with my names and then the various, however I wanted to leave it. Now there are a whole bunch of cases that are not reported, about 14. If I don't want to see them, I can go back and I can customize my bin again. I can say, okay, the missing ones, and then the not reported ones, hide them. And those move down here. And I can do save bins again. So now it's plotting, the stages without any data that I don't have data for. So, so that's a neat feature. And I can actually, print it as an SVG. And it's showing me, it did that for me. I mean, downloads an SVG. Chrome can open it up. If I want to go back, I can edit it. I can add these back in. and I can create another category for like other or something for it. Or I can go back and ungroup these however I want to. So I can also go back and reset them, reset the default, and goes back to where it was. So that's a categorical binning. So we'll do another feature. So I'm going to go back up and clear my search. I'm back to where I was before. And let's say I'm just moving down, and you can move down however you want to. In this case, we have age of diagnosis, but I'm also interested in year of diagnosis. Let's see what we have for year of diagnosis. So the year of diagnosis is an interesting data, and it tells us, the time period when the initial diagnosis was made for the patients within this cohort. And as you can see, there were about 5 cases from 1978 to 1985, and about 13 from 85 to 92, and so on. And it also tells us, about the knowledge, about our, better awareness, understanding of the disease. So as people have learned more, being more aware of skin cancer and exposure to sun and stuff, they're going to the doctor more often. Even doctors are checking themselves, when they're in physicals. So as you can see, the awareness, with the awareness, the number of cases has also increased. And that's, I would say that's important data. Otherwise, it skews some of the, statistics, can't skew statistics. And same thing, I can do survival plots for these. Another thing I can do is, I can also customize this data. So when I go here, because this is numerical data, it's telling me automatically, it's giving me that it divided the data up and into intervals. So it's giving me a default of 9, and it divided up from 1978 to 2014. So if I just take the default interval and save bins, it plotted into 4 bins. So it basically, the default will break everything down into 4 quantiles from 5 to 4 now. And it's giving me 1978 to 87, so 4 even bins. But, these kind of look skewed to me. So I'm going to change them. I'm going to go back to edit bins, and I'm going to make them 10. So good decades, and see how it looks. So it gave me 1978 to 88. So it's fairly good, and it's telling me the range from 78 to 2014. I'm going to go back and change it again. So however you want to, there's no rules to it. I'm going to play with the values over here. So I'm going to do it to 1970, 2014, and do again. Now it gave me nice decades from 70 to 80, 80 to 90, and now I can see there was 2 cases because the first case started in 78 within this cohort, and this gave me, and I can go back to my histogram, and it plotted the histograms, and it's telling me how the number of cases when the year was diagnosis. I can also do, look at survival for them. If the number of cases is too small within the group, you can plot it, not enough data, because we can't calculate the p-value for that small of a number of cases. So you can group them again if you wanted to. So you can So I'm going to do something else. So I'm going to show you the custom range feature for this. So if I go to custom range, I can create now my own bins. So I can do is whatever name you want to do it. You can give it a bin 1. In my case, I'm going to give it a pre-2000, and I'm going to do 1970 to 2000, and then post-2000. So if I go higher, it's going to come back and give me an error. Come back. I do add. Now I have two groups for it. Give it two names, save it. And now it actually created, two different bins for me and added them all of them up. So I have 78 cases, pre-2000 year of diagnosis, and then 389. Other thing I can do is, besides this, is I can create a cohort for each one of these. So I can actually do it, click on this, and create action, and I can create, save a new case, and it's telling me a custom name, so I'm going to just call it KCM pre-2000. Uncheck it, do the other one, and so it created two other. We'll get back to that where it went. So one thing you can also do it is, if you wanted to eliminate these pre-2000, I'm just giving the example, from your cohort, I can check this, and I can do is remove from the existing case set. So when I click on this, it's telling me, so there are the two other ones I created. Within this, I want to remove the pre-2000 cases. So I started with 470. I confirm it, yes. And it went back. So now I have is, it's showing me my diagnosis. So it came back, and it's plotting the, so I removed it. So there's not enough data for me. Oh, it removed the other one. Let me come back to that in a second. Oh, I deleted the larger case. So my error. So those are the different things you can do with it. Once you have created your cohorts, you can actually go back into repository or exploration and view them. So in this case, when I created the three, I can do is upload case sets. I can click here, and those would be my my cases. In this case, I'm going to go back to the post-2000, and I'm going to click on that, and it's showing me, and I can create another cohort, or I can go into repository, and I can download those cases, data for those cases. I have files for those cases. So there are many things I can do with them. Going back to my analysis. So if I don't want to create a new analysis, run an analysis, I can go back to this results tab, and it'll show me my previous analysis. So in this case, it's showing me the one I just ran, and it kept there. I can actually copy the analysis, and I can change my analysis to a different, in this case, the one I created accidentally was a smaller one with 3. So if I go back and create another one, I create those cards, showing you the last one. I can go to brain, and we'll run that. So basically, using those cards I had, now I can actually go back and run a different cohort within them. So there's no staging data, pathological staging data for this cohort, so I can remove that, and however. And you can see it's kept my bins, the custom bins I had created. So this is kind of very powerful feature. What else can we do? Oh, let's go down, scroll down, and we'll see what else. So there's same thing. If there's days to treatment, I can go back and it'll tell me days to treatment data information for it, etc. And those are green. I can go and do exposure, see if there's any alcohol history data collected for it. There's not much. Most of it is missing or not reported. There are some cases. In this case, I can do smoking status, the packs of cigarettes and stuff, and not that much for it. I'm going to remove it. Let me try BMI. So it's giving me BMI for this cohort. Now on brain cohort, I think I changed it, yeah. This is kind of interesting too, because we're collecting the raw value, the BMI value, for each of the cases. And BMI is something that can be categorical or continuous. And I can use my binning again to actually make it. So if I wanted to make custom bins for, underweight and overweight, I can do it from 0 to 18.5. Normal weight BMI, I can do it 18.5 to 25. And overweight 25, and then the max value is 47. So if I add 48, I think it'll give me an error. Oh, it'll let me do that. I can hit 47. I can do save. Now, it actually converted my continuous into a categorical if I wanted to. So it's showing me, the values. So, people like me, myself, I'm in the overweight category, so I'm in this category. So, but it's a good feature to actually, if you wanted to organize your data. What else do you have? Show print. Go back up. So there's this print feature. You can go in. It'll print you the cards for you and basically save it as a PDF on your browser and on your desktop, wherever you want it to. I can actually go and get the values. So I can get these and save them as CSV. I think I showed that. And it's showing me them. And I can go to each one of them and it'll show me the real cases associated with it in this study. So those are all very useful features. The idea is you start with exploration, create a cohort, go into analysis, and circle it back into exploration and others. So that concludes my demo. I'm going to go back to my presentation and make sure I covered everything. So I think I showed you how to do the create a name, default name, and cases. So I just put it together so that it's useful sometimes to just also look at the presentation quickly as a quick check. So as you can see, there are many of them you can do. So we went through the different things. Search, clinical cards, custom bins, binning, copy, download, print PDF. Back to exploration. Oh yeah, there's one more feature. Let's go to manage sets. So I'm going to go back and show you. So this is one way of exploration, is one way of creating your cohorts. Another way is to go to manage sets. So in manage sets, you can actually, it's showing you all the sets that I created, and I can manage them. I can download those those case IDs. I can view them in repository. Like when I click here, view files in repository, it's showing me those within the repository, the cases, and the files associated with them. And I can add them to my card, download them, or use the DTT to download. So those are all very good features. Going back to manage sets, I can also upload my own cases. So if I've gone through analysis and have a set of cases that I'm interested in, I can actually add them here. So I can do is upload sets, click on that, and then it's telling me I can upload cases, genes, and mutations. Those are the three things we support. So I'm going to start with cases. In this case, I have to give it a name. So I'm just giving it my cases name, but you can give it a more descriptive name. I can type in the case IDs over here as a comma delimited. And as you can see the example, they can be submitter IDs. Those are the study-based submitter IDs, or they can be UUID. So you can have a mix and match of those. Or you can upload a file. So you can browse, you can upload a file. So going back to it, I have a quick example. I just copied some submitter IDs, and I actually made some errors in them, added some errors in them. So in this case, I'm going to paste them here. As soon as I pasted them, and same thing happens with your browse data when you upload a file. I passed 3 IDs, and it told me it did a mapping of them. That this case UUID maps to this is my case ID, and it's from TCG Read project. And then the second one is mine. It's showing me. And then this is the one I made a mistake in. I added a number to it. And it's telling me that it did not find that ID. So this is a very cool feature. And I can now submit it. So there are my cases over here. It only took 2. So I can go back into exploration and use that as my input. So I just did 2, but you can do the same thing for analysis. So it's showing me, the 2 cases, and then I can go back to over here demo and do the same thing. So there's a user guide where the functionality I just showed you in the GDC user guide. There's a page on that that describes all of that in detail, really good examples. There's the data model page that describes the data model. And the reason I'm showing the data model is because the clinical fields are based on the GDC data model and also the dictionary. So those are 3 all important links if you need more information on any of the clinical fields. And then I have some references to the GDC website, documentation, portal, etc. That's my last slide. So Sharon, are there any questions?
Yeah, there are a few questions, Himanso. The first question, we got a comment that appears to be a very useful feature. Am I able to create or refine a cohort, and are these features available through the API?
Refine the cohort. So these features, I believe, are available through the API. I don't have an example at this point, but I would say it's contact user support, and we can probably point you to what the examples are for them. I'm sorry, I didn't prepare for the API examples.
I think to also on that question about creating a cohort, I think was further refined as, am I able to create or refine a cohort by passing in a list a subset of patient identifiers?
Yes, so you can do that. So basically, what I just showed over here in Manage Sets, so that's how you can create it. Now, this functionality is within the portal. It is not supported by the API. So once you pass, so the portal is actually performing the same thing what the API does in the sense that it takes the list of user IDs, submitter IDs, and sends it back to the GDC through the API and validates and brings back the results. So that would be same as using any the API and passing the list within the API. There's no special any other feature for that. Bill, do you want to answer that, or I don't know if Bill's online? That's my understanding of it. Sharon, did I answer it? Question?
Sorry, I was on mute. Yes, you basically covered it, that these features are kind of meant to be used through the portal, but various parts of them can be used through the API. And if you contact the user services help desk, we can clarify that further.
Okay, thanks, Bill. There's a question on, could you tell us what technology, that is R, Python, et cetera, is being used to create the graphs and charts?
I'll leave the development team answer that question. I believe it's mostly JavaScript, but
yeah, I mean, we do make a library available on OncoJS. It's JavaScript library that's actually reusable, and we do make that a link off of our GDC website to the library. So you could actually reuse like the survival plot and other plots that we do if you go to OncoJS, and there should be a link for it on our GDC website under development.
And as far as the other technologies, also, if you send it to user support, we can definitely give you more specifics too for that.
So we have another question. Once we create a cohort based on clinical criteria, can we download other data like the imaging and possibly any of the omics, the genomics type data?
Oh, yes. I didn't cover that part of it since I was concentrating on the clinical. But once you've created, that's the whole idea is, once you've created a clinical cohort, then you can go and look at the genomic data for it and do further analysis. So, for example, I am going to go back and look at the one I messed up. I'm going to go back and look at that as my case. So I'm going to start with that. And it's telling me, I'm going to actually start clear and then start again so that you can see it better. So if I do here, and I'll take this one. So it's showing me 467 cases, and these are the genes associated with it. This is the open data, and then it's just gene analysis, and I can download the gene sets based on that. I can do a mutation, look at the mutations. I can go to OncoGrid and look at the mutations and the copy number changes for that cohort. So it's all kinds of, I can do analysis for it. I can also do other analysis. I don't have it created right now, but you can do cohort comparison. I can do a select, and I can pick the two cohorts. In this case, and it's telling me between the two cohorts, what's the gender, vital status, age of diagnosis, etc. for them, and I can add ethnicity, and it's telling me what is the subset or superset of them, between the two, what's the Venn diagram for them. And as you can see, it's back to my results. I can download it, and I can do, other explorations with So that's what I was trying to get back to my slide.
Yes, and in both these, the slide images, the diagnostic and the tissue are available for download as SVG file. Are available for download on the repository as well as all the genomic data. But the area you want to go to for download is a repository.
Right. So the same cohort, I can go to repository. It's showing me over here what are the slides associated with it. So I can click here, and it's showing me the slides for that case. So, and I can actually go to the repository, and I can download them too. So, these slides. Did I answer that question, Sharon?
Yes, yes. Thank you, Himanso. How about where can we get a list of all the clinical data elements that the GDC supports?
That's a good question. I'll go back to the GDC dictionary, and the dictionary is where they all are listed. So if you go to case, under case project, so it shows you the association, and it shows you all the things that are listed for it that we accept. So dictionary is the best place for it. There's another site within the dictionary search, another feature, part of the data dictionary. The viewer shows you all the raw values, and you can, every value. But there's another tool within the dictionary called search, and that actually lets you pick. So if you wanted to do your mapping and you wanted to search on something, it tells you, you can put in anything and it tells you if it's a value, property, or dictionary. In this case, it's a disease. I can click on it, and it's telling me, let me go into details, show all the values for it. It's giving me the property. Click on here, and showing me, the synonyms for it within the various EBS, NCI dictionary and stuff. So they're all tied together. So basically, every term we have, you can go and search synonyms within the data dictionary within the search tool. The other place you can also look at all the values is, I'm going to go back to analysis, to the data portal. I'm going to go back to analysis. So we had thought about this for a while, and did we want to show, all the different things we support? So if you go down here within analysis, so within cohort building, you can filter out things that have values or things that don't have values. By that, I mean is I can go into here, clinical, and I can see show fields with values, so 52. If I uncheck it, it's going to show me all 95 fields. And some of them may not have values, but I can actually see all the things that we collect. And once I check it, it reduces it. So it's a feature to help you create your cohort in a faster way, but showing you all the properties that we support. Similarly, within the analysis, is doing the same thing. That's why we didn't actually, close the ones which don't have values. So if there is no cause of death data, in this case there is some, but if there is none, then we're not stopping it. But you can see all 59. So you can go through it and discover all the things that GDC collects, clinical properties that GDC collects during submission.
Okay, we have another question. Can we make our cohort converged on a clinical criteria? Can we make it public, maybe with some identifier such as a DOI?
Well, the clinical data right now in GDC, all clinical data is public data. So it is, it's open data. So it's not a control access data. So I don't know why. I think the answer is yes. Bill, you want to answer that? I don't want.
I would agree that the answer is yes, but I want to bring to the user's attention that this won't always be the case, and in the future we do expect to have some projects with controlled clinical data.
Okay. All right, it looks like that was the last question in the Q&A area. Let's see. Actually, I guess a follow-up, can that data set have its own DOI is kind of the question. I guess one more follow-up from that previous question.
So we're supporting UUID. So if this is, let's say, I am looking for within brain mail as my data set, I can just click here and I can look and download these cases, and these cases have UUIDs. We're not supporting a pure DOI in the sense, but we support UUIDs, which is a much finer grain than a DOI. So yes, so you can, once you have the cases, that's your cohort, and you can use it within your research, your publications, and stuff.
Okay, if there's any other questions, please place them in the questions and answers. I guess as a follow-up to that, the DOI for the cohort itself, so sort of the entire cohort that was built using the query.
Right. So we don't have a DOI for the cohort. That's an interesting idea. So right now, once you create a cohort, you can export it, and that's giving you all the IDs for it. And if you wanted to, we're not managing that as a DOI. That's why I said is we're not giving you a cohort level DOI. But if you wanted to manage yourself and create a DOI for it, you can certainly do it on your own. But, so we do allow you to download that cohort and use it however you want to. So, but within GDC, there is no feature or functionality to persist it as a DOI.
Okay, and there's a question, do the custom cohorts we create still disappear periodically?
Yes, they do disappear. That's a good question. So it says that the custom cohorts will be deleted. So when we do have a data release, and this is from the NIH policy, we can't really persist them. We can't really hold, user data within GDC. So it's basically your browser that's supporting this is actually capturing it. And once you clear the cache, or when we have a new, release, we clear it out, and then that's, it goes away. So that's why it's always a good idea to use this manage sets feature and download, once you create a cohort, download these UUIDs, and then use them, give it a name, or, manage them yourself. And you can always upload them back in. So you can use, once you downloaded them, you can use the upload feature to bring it back in. Whatever you downloaded.
Okay, here's a question. From the demo, the survival rate goes down in recent decade comparing with the previous decade. Why does it happen?
Survival rate, I can't really comment on the data itself. That's a very specific question about the data. I would suggest is that go to asset and user services, and we can research it specific for the study or cohort that you're asking and answer that. I'm sorry, I'm not a researcher to answer that question specifically about the data. We can try to find you an answer.
Okay. All right, looks like there's no other questions in the Q&A. So we will adjourn this webinar, and it will take us a little bit of time to get this the video recording transcript updated and get this on our website, but it will be on our GDC support area of our website, hopefully in the near future. Thanks everybody very much for attending. Have a good rest of the day.